Hello, Master Gardener trainees. I just want to take the opportunity to introduce myself a little bit before we get started today, and also say welcome to the Master Gardener family. Very excited to have you. My name is Whitney Elmore. I am the County Extension Director and Urban Horticulture Agent and Master Gardener Coordinator in Pasco County. I want to give a shout out to Dr. Bill Lester for asking me to do the plant pathology introduction for you today for this segment of your training. I have to say that plant pathology to me is very exciting, huge, very diverse field within the sciences. It's kind of what drew me to it to begin with, because it does involve not just the science of plants, but it involves fungi and bacteria and viruses and nematodes and weather patterns and soils, all different kinds of aspects of the biotic and the abiotic world that plants could come in contact with. And we're oftentimes studying how those things interact with each other. So we're going to be throwing around quite a few terms. Plant pathology, like I said, is a very diverse field. It's big, it's huge, so we're going to do our best to break it down into its smallest component, take out a lot of the complexities where we can, so that we can give you tools you need to get started as an amateur plant pathologist, say working in the Master Gardener Plant Lab, for folks coming in that need help with a variety of things. So there's going to be quite a bit that we're going to study, but I think and hopefully everybody will be able to pick up some key skills by the time we're done with this particular presentation. So just a little bit about me. Prior to coming to Extension in Pasco County, I was a professor up in Georgia. So before you hate me too much, I am not a bulldog. Sorry for any Georgia fans in the room. I am certainly a gator. I am originally from Kentucky, went to Western Kentucky University, where I have degrees in agriculture and turfgrass science, and then came to the University of Florida to get my doctorate in plant pathology. So I'm absolutely a hilltopper and a gator. So let's go ahead and move on and look at our objectives for this lesson. First of all, we need to talk about what disease actually is and what it isn't. It's just right off the bat, a lot of folks are somewhat confused, and it seems like one of those terms, at first glance you go, well, we throw that term around all the time. So obviously I know what it is, but when you ask somebody to define it, they usually start to stumble and have a little bit of trouble defining the particular term. It's kind of like asking somebody to define life. I used to do this to my students a lot when I taught or tortured students. I would ask them to define life, and they go, well, life is life. But any good English teacher will tell you you cannot define a term using a term, and that does not explain and give a definition. So we're going to come back around to the word disease and what it means. That's our starting point. We're going to go over a very important concept in plant pathology, which is the plant disease triangle. We're going to expand on that just a little bit to talk about some of the factors associated with disease in plants. Just a smidge about disease development. This is where we can really start to get very complicated into how plants respond to both living or biotic factors and non-living factors or abiotic factors in their environment, and whether they're going to become diseased, if they're going to get a deficiency, become disordered, or if they're going to live out just a perfectly healthy life. There's a lot of factors that come into play, and plants are quite fantastic living beings. Just like animals have a system, an immune system, plants have what I consider a variety of immune systems. Not even so much as systems, but ways to counteract all the different things that nature can throw at them. In some ways, in my opinion, a little bit more advanced than what animals can do. So plants are quite good at dealing with disease, even if they do get quote-unquote sick. So we're going to talk just a little about disease development, but not go too far down that rabbit hole. It's a pretty complicated set of concepts that I think would be beyond what your use would be and probably your interest as well. Talk a little bit about fungal pathogens, bacterial plant pathogens, viral plant pathogens, nematodes, and then a little bit about disease management, which is where I think you'll spend a whole lot of your time in dealing with homeowners especially. So plant disease. Plant disease, the best way to define disease, whether it be in plants or animals, it doesn't really matter, is it's a malfunction in the organism. Any malfunction in the organism is a disease. So that malfunction can cause changes in the plant appearance or in yield loss. It can cause the fruit to become distorted or maybe not fruit at all or drop the fruit too soon. It can cause a wide variety of things, all the way from issues that we would probably never notice with the naked eye all the way to death, fatality in that particular plant. So in your gardens, in your home lawns, around the house, your house plants, this might not be a huge issue, or it could be a very serious situation if you've invested a lot of money. But go over onto the agronomic side of things, where we're producing food for the rest of the world, and the difference in plant loss or yield loss and plant appearance can be very drastic, and it can mean the difference in some cases of a farm closing down all the way to potential for famine. So it can be a very serious situation. So plant disease doesn't have to, in not all cases, cause death. There is a variety of potential causes that all these different organisms that we're gonna talk about, and we can't get too specific because we have so many different types of pathogens out there, but we can generalize them into categories like fungal, bacterial, and viral, because they all tend to behave and appear in very similar ways, and we would also try to combat them in very similar ways. And you'll see what I mean when we come back around to that. But a variety of potential causes to why plants can get sick and die or have differences in appearance and behavior. This could be because a pathogen can directly damage cells. So cells are the smallest units of life. And if you start to damage and kill cells, you're obviously on a small localized level going to have a reaction that can translate to a bigger reaction, systematic across the entire organism. You have some pathogens that can produce toxins, which can obviously do damage or cause death. You have some that can produce growth regulators that can make the plant get bigger or smaller, or the fruit bigger, smaller, distorted, can do all kinds of weird, almost fantastic things in some cases. Then there can be other byproducts. Just because of how the plant is having to react to the pathogen, that plant's going to put off byproducts. It's a living being, and it's going to produce chemicals in reaction to being attacked by a pathogen, and that can affect itself how the plant will behave, just because of the different things that it has created during its metabolism. A lot of times we see disease organisms in plants affect the use of nutrients, the use and uptake of water, or their interference in some cases. And those are some of the things I think you would be much more readily recognize, and usually a little bit more quickly recognize out in the field. A couple of concepts that I do want to bring to your attention. I did mention the terms biotic and abiotic, so make sure you're familiar with those terms. Living and non-living factors that can affect organisms. So from a biotic standpoint, plant disease can be caused by bacteria or fungi. Those are living entities that can cause disease. What about on the abiotic side of things? You throw an A in front of a word and the Greeks will tell you that pretty much means not. So not living factors that can cause disease or disorder or predispose them to getting an infection with a living thing. So think about water. Too much, too little. Water's not a living thing. too little sunlight. You put a shade-loving plant out in full sun, it's going to be weakened and it's going to be predisposed to disease or disorder. What about compacted soil or soil where we have a pH that's not conducive to the uptake of nutrients? So those are abiotic factors, and there's many, many more that we'll come back around to later on. Before I move on, I do want to make one quick comment here. If you hear any kind of rattling, if you hear any kind of barking, that's not me having some type of reaction, hopefully, to giving this lecture. I've got 3 furry babies, 3 dogs running around in here, so hopefully everybody will behave while we do this. So forgive me if there's an odd sound back here. No matter what it is, I'll blame it on the dogs. A couple of other concepts I want to bring to your attention, and these are the types of things I would suspect you would see on a quiz or on a test as a master gardener. The difference between signs and symptoms. And no matter who I have taught this to, from college students all the way down to kids in high school or middle school, master gardeners, folks seem to have quite a bit of trouble with signs and symptoms of disease. They are not the same thing. We tend to find ourselves using those terms interchangeably, but they are not the same. So what's a sign and what's a symptom? So let's say somebody brings into the plant disease clinic a chlorotic or yellowed plant. It's laying there in front of you, and all you see is this bright yellow plant that should be a deep green. That's a symptom of a problem. It's something I can see that's tangible, showing me there's an impact on this plant from something. Whether it's biotic or abiotic, I don't know right now. But it is a symptom of an issue with that plant. Now, what if I start to look under a microscope at that particular plant leaf and I find this web of mycelium? And we're going to learn in a little while that that's the body of a fungus. What if I see symptoms and then I see fungal body associated with those symptoms? That fungal body is a sign that there could be an infectious agent. So if I can see, and sometimes you can and sometimes you can't, if I can see the actual body of the pathogen itself, and a pathogen's anything that can cause disease. If I can see the body of it, that's a sign that it's there. If I can see the result of the impact or the malfunction caused by that living thing, that's a symptom. So signs and symptoms are very different things. Let's run back through here another example before we move on. So I go out and I find in my garden that all of my, cucumbers are stunted and kind of distorted. They're very small and they've got all these spotty flecks, almost like a mosaic on the leaves. Plant's just not doing very well. It's a surviving and it's making it, but it just doesn't look very good. So I noticed distortions, twisted discolorations, and I noticed stunting. Are those signs or are those symptoms? Those are going to be symptoms or malfunctions caused by some biotic or abiotic factor. Now, as I start to look closer, I might look for signs of fungi, let's say. But what if I find none? No signs, no fungal body. What if I look further and do some diagnostic tests to look for microscopic organisms like bacteria or entities like a virus, but I find no particles or pieces of those particular entities or organisms, no signs? Would I be able to then say that those organisms had some sort of impact on that plant, caused those symptoms? More than likely not. We usually have to find signs and symptoms together when we can to identify the actual cause of a disease. But you can definitely see disease symptoms without seeing signs, when you're talking about dealing with microscopic beings. Your naked eye is not going to see a bacterium that's causing a symptom of disease. Could it potentially see some fungal bodies? Yes, but not bacteria, and certainly not viruses. And we'll explain why a little bit later on. So, very important concept here, kind of the crux of plant pathology is what we call the plant disease triangle. So you see the three different points here, the host, the pathogen, and a favorable environment. For disease or a malfunction in an organism to occur, all three of these factors have to be in place at the same time. Same place, same time for disease to occur. The good thing here is that disease is not the norm. And that is a really good thing. If it were the norm, we probably would not exist at this point in time. There wouldn't hardly be any living thing existing if disease was the norm. It's the exception because you have these three factors that must come into play all at the same time. It's kind of like the perfect storm. To get disease, you must have a susceptible host. You may have a particular cultivar, you may have an entire crop, but you have to have a host that is susceptible to a particular pathogen in a favorable environment. Not favorable for the plant, but favorable for the pathogen to maybe produce more of itself or find an entry point into that plant. You've got to have a favorable environment for that pathogen to survive, find its way into the host, and make that host sick, basically. So favorable environments, air temperature, soil fertility, soil type, soil pH, rainfall, humidity, soil moisture, so many different abiotic factors that can come into play here. The pathogen. You have to have a pathogen capable of causing disease in that specific susceptible host. The other good thing about diseases is that a lot of pathogens are very host specific. So you might have a particular bacterial pathogen that will only attack one cultivar of a particular type of plant. And if you remember, go back to what a cultivar is. It's a particular type of plant that we're cultivating for a specific reason. So we might have a certain type or cultivar of tomato that's highly susceptible to a certain bacterium, but not all tomatoes are. That's why we can kind of take advantage of those concepts and just not plant that cultivar anymore. And we wouldn't have that same particular disease if that particular bacterium was common in that area. So we can kind of play against these 3 points in many cases. We try to take at least one of these factors out of the equation, and you can even take the host out of the equation but still have your plants, and we'll talk about how we do that. But you remove one of these 3 points and you are going to greatly reduce the likelihood of disease. Now, I'd like for you to stop and think if there might be another aspect that could fit in here. And instead of having 3 points, maybe we have something more like a square. We're starting to expand our knowledge of plant pathology at this point because that helps us in preventing disease. I'll tell you right now, curing it is downright impossible. Prevention is the key, not only in animals, but certainly in plants, whether it be agronomic or in the home garden or house plants. Preventing is the key, not trying to cure. We're not going to cure plant diseases. We might be able to stop them from spreading, but we're not going to cure. So what's a fourth component that might come into play here? I like to include the concept from Maz, which is time. We expand this triangle to include time, and with time then, if you stop and think about it, when is the best time to plant, say, your oak trees? When's the best time to plant your tomato plants and your pepper plants? How many folks have come to Florida from up north and are so frustrated with the fact that it's hard to grow plants or tomato plants in the summer, and they have not realized that they're at the wrong time of the year trying to plant. Summer is not a growing season for us. It is in New Jersey, It is even in Tennessee in some cases, but it's not for us. So time of the year, season is a factor, and also when we're putting out plants in terms of their age and their ability to tolerate certain environmental conditions and even biotic conditions that might come into play. So time is a fourth component that needs to be considered very heavily here. Also, think about the fact that we have a rainy season and a drought. Those are normal aspects. We have a dry season and a rainy season. If you have particular types of plants that need a lot of rain to get them established, why would you plant them in the middle of the dry season? Okay? So we can start to pull apart different aspects of this triangle, even the expanded triangle, to make sure we keep disease the exception and not the norm. Can you think of a fifth potential component? I sure can. Humans. Humans, absolutely. And of course we can affect time and when we plant something. We can also have an impact in the types of plants and cultivars we put out. Even the environment. Now you may go, now how is that possible? I can't affect when it rains and when it doesn't. But what about if I've got plants in a greenhouse? I can certainly control the amount of moisture and the temperature in those types of settings. What about using drip irrigation or micro irrigation instead of overhead irrigation? I am controlling the environment in those particular cases. What about the pathogen? Can I control those? Can I cause there to be more of a particular pathogen, meaning more potential for disease? Can I make there be less? Absolutely. So human activity is going to have a big impact here. So I always like to consider that as a fifth component. And I'll point out some places a little bit later where that's very, very relevant and might surprise you a little bit. Alright, so let's start to talk about some of the different factors. Let's pull apart that initial pyramid and talk a little bit about the host factors that we should take into consideration when thinking about disease in plants. All plants are living things, so you really should consider all plants to be a potential host for some sort of pathogen. There's pretty much something out there that can attack most anything else that it can find. There are some plants that tend to be what we might say bulletproof. That doesn't mean they're not exposed to plant pathogens. They may just not get sick with them. Or they're able to tolerate the infection to some extent for a variety of reasons. Some plants are notorious for not being able to tolerate much plant disease pressure or insect pressure or even weed competition without it taking a tremendous toll on them. So all plants, even the hardier plants, can be considered host of some sort of pathogen, whether it's common or not, doesn't really matter. When you think about crop plants, or a lot of the plants that we usually put out into our gardens, we're monoculturing. And that's done for a variety of reasons. It's very beneficial when you're wanting to make sure you have uniformity and marketability of your fruits and of your vegetables. But also in how we are going to be planting those and harvesting those. We have particular types of machinery that need to have certain types of, or the same similar types of heights on plants and whatnot for those machinery to run through there productively and efficiently. Lots and lots of different reasons that are all very good intentions of having monocultured crops. But one of the big drawbacks that we have figured out finally in the last 30 to 40 years here is that monoculturing predisposes us to some pretty drastic plant disease situations. If everybody's identical out in that field, if they're all clones, they're all genetically the same, they're uniform, they've been cloned, and if one of those plants then is susceptible to a particular pathogen, because they're clones, they all are. So we can have an entire crop, not just in one field or two fields, but an entire region, an entire country, in some serious situations wiped out very quickly by one particular type of pathogen. So monoculturing has definite benefits, but it does have the drawback of having no genetic diversity. Genetic diversity in all living things gives us a potential to adapt or change slightly. That's not always the case, but there's a potential for us to be able to handle disease pressure and survive. We might not look the best on the other side of it or perform the best, but we might be able to survive or get a crop out, have some foods to eat, or not lose the entire breeding line completely. The idea that it could go extinct exists out there. So that degree of genetic uniformity certainly plays a factor in hosts and how vulnerable they are to disease pressure. Age, I think we all have started to probably realize that age plays a definite role in our lives, so it would in plants as well. We know that younger plants are typically not capable of dealing with plant disease nearly as well as older, more established types of plants. So older plants usually can withstand some disease pressure, depending on the interaction and the organisms involved there, but generally they're better, more established and capable of dealing with pathogens than young ones. Think of maybe having some small little seedlings of tomatoes in a tray, and overnight they look great on one day. Overnight you come back in and they're all down. Where you've lost all of them overnight, maybe to a fungal infection. So the younger ones usually don't hold up quite as well. Hosts, plants even have different levels of susceptibility to plant pathogens. And again, I don't want to go too far down this direction because it can get pretty complicated, but you may see some of these terms in reading that you do in articles or EDIS publications, even on some of the information packets on seeds or even the little labels on plants when you pick them up at the store. Terms like immune. Immune, just like the way we would think of it in the animal kingdom, a plant cannot be infected by a particular pathogen if it's immune. Doesn't mean another pathogen couldn't get it, but a very particular pathogen, okay, that's host specific, wouldn't be able to attack and kill a particular host plant. We'd say that plant's immune. But what if it can be infected but not become diseased? And there is a definite difference. We can become infected but not diseased. So think about if you were to get, and we'll go a little on the gross side here, if you were to get a ringworm infection. That's a fungal infection. If you were to get a ringworm infection on your arm, does your entire arm become diseased and malfunction? No. Were you susceptible to that ringworm infection? Absolutely. Did you have some abnormalities or some impact because of that? Yeah, you probably did. But you did not have a serious malfunction in the use of that arm. So some plants can be susceptible to a particular pathogen, and they can become infected even, but not become diseased. And we'll talk about the differences in just a minute. Other plants we say are resistant. Resistant means it may or may not become infected, but it's not going to become diseased down the road. And these types of host factors allows us again to possibly remove the host equation out of that disease triangle. We've got our environment, we've got the pathogen, let's say, that's widespread. But what if we can plant a host that's immune to that particular pathogen? Or one that's resistant to that particular pathogen? Maybe it becomes infected, maybe it doesn't, but it's not going to be killed. It's not going to become malformed. It's not going to malfunction and become diseased. So you may see these types of words on some of the plants that you pick up or some of the seeds, and it gives you an indication of the fact that we're taking advantage of the fact that particular pathogens are not going to have a serious impact on that particular cultivar. Pathogen factors. So inoculum. What does inoculum mean? Inoculum is really the pathogen itself. Okay? So let's say I had a million spores from a particular fungal body. That's all inoculum. Pieces of the pathogen that are capable of causing disease, infecting a host and causing disease. That's the inoculum that we're dealing with. Particles of the pathogen is the way that we'll put it. And any of those particles could pretty much cause disease. The more inoculum or the more pathogen, the more potential for disease. I think that only makes sense. The genetics of the pathogen certainly comes into play when we start talking about hosts being susceptible to that pathogen or not, and even the virulence of the pathogen. How serious is that pathogen about being able to cause disease? Now, it's not a conscious effort. What I mean by that is what types of mechanisms does that pathogen have to get into the host, to colonize the host, to infect the host, and to cause it to malfunction or become diseased. And that varies greatly. What about the type of reproduction of the pathogen? Does it have one cycle in its life of being able to have offspring, or does it have many? So mono meaning one, many cycles of producing offspring, or with monocyclic, or many cycles, polycyclic. That's obviously going to play a big role. The more cycles of being able to have babies, the more babies, the more inoculum the pathogen could produce. The ecology then. So where does this pathogen prefer to find itself, and how does it spread itself around to new hosts? Remember, these are, for the most part, living things. And its job as a living thing is to perpetuate itself, to keep its species around, to keep having babies. Find hosts, find suitable living space, habitat, food, moisture. So is it moving itself around in the air? Do air currents play a role? Is it found in the soil and moving through the soil? What about in water? We even have some fungal pathogens that can swim. Is it moving itself around in infected seed? And what about vectors? A vector is an organism that's capable of moving around a pathogen to a susceptible host. So in Florida, we've got serious issues with citrus greening. Most everybody knows that these days. That is caused by a bacterial infection. That bacterium is moved from plant to plant by a vector, a psyllid, a tiny, tiny, tiny little piercing, sucking insect. That's the vector. So is the pathogen dependent on a vector? Some are, some aren't. Is that vector common in an area? Does it exist at all in an area? What's the ecology of the vector? You get into some very complex relationships here outside of even the plant itself when you get over talking about pathogen and vector relationships. Environmental factors. I think most of us recognize that moisture certainly plays a role. We oftentimes think of having way too much moisture as causing an issue. Sometimes having too little can cause an issue as well. Temperature. Hotter temperatures, sometimes cooler temperatures, it all varies, can play a big role in disease development. How happy is the plant and how happy is the inoculum of the pathogen? That's going to be dependent on moisture and temperature. The effects of our human activities. Are we monoculturing? Are we reducing the genetic diversity of the plant's natural ability to defend itself, basically? Are we creating more inoculum unknowingly? Are we lowering the quality of seed and actually have seed that's infected and we're spreading inoculum this way? Are we leaving behind diseased residues in our gardens? Pieces of plant material full of inoculum of a pathogen that's just waiting for us to plant another plant of that same cultivar in that same spot, which we tend to like to do, and we then give it a food source. Are we rotating or not rotating our crops? Are we rotating our gardens when at all possible to reduce disease pressure in a particular area? And are we using alternate hosts? And what I mean by this is maybe planting cucumber in a certain area of your garden where you've been planting tomatoes for a couple of years. Move outside of the species and certainly outside of the genus. So don't come back in with closely related plants in the same area, especially when you've had disease problems in the past, because you're probably going to just have them again. And is there potential we're introducing new pathogens? And we can do this just by bringing home plants from the store. We could do this from driving our vehicles through one field and over into another. We could do this on our hands. And we can certainly do this with seeds. And with global trade and travel at this point in time, introducing new pathogens is commonplace, unfortunately. Just a little bit about disease development, and I have hinted at this a few times here. You have a series of sequential steps that lead toward a malfunction or disease in a plant, and anywhere along the way, you can have plants that are resistant or immune that are capable of stopping this process and preventing disease. Remember, all plants get exposed to pathogens. It doesn't mean they become diseased. They have some miraculous mechanisms to stop some of these steps in action to prevent the progression toward disease. So, general order of events in disease development. What has to happen and generally when before we get disease? First of all, you got to have the pathogen get to the host. Did it get vectored there by an insect? Did it get washed there during a rainstorm? Did I carry it on my hands? Or was it in the seed? Many potential issues there for dispersal of the pathogen to the host, but to get disease, you've got to get the pathogen to the host in the right environmental conditions. Second thing that has to happen is that pathogen has to get in to that plant one way or another. Now, when you're talking about fungal pathogens, fungi have the ability to penetrate their host. They can either do this through pressure, using their bodies to force their way in. They could use natural openings and just grow into those natural openings. Or in some cases, they can produce toxins that break down the epidermis of the plant, allow for a sore, and allow for the organism to grow into and then begin to infect or spread into the plant. Bacteria and viruses are different. Neither of the two have the ability to produce toxins to break down plant tissues to get into the plant and penetrate it. So how do viruses and how do bacteria get in and cause disease? Natural openings and wounds, just like with animals. You got to have a wound and you got to have that pathogen get into it, or you got to have a natural opening. So on plants, you think about the nectaries, different parts of the flowers, the margins of the leaf, the stomates. All of those are areas that are open spots leading directly internal into the plant. Potential for then those types of pathogens to get inside. So penetration and infection of the host, where the pathogen starts to move throughout host tissues, then it becomes invaded. So you've got the tissue where it originally infected, but once you get a movement outside of that original tissue, kind of like a cancer, if you will, you get an invasion. An invasion of the pathogen and then colonization of the host. It's almost as if, and in some cases, it takes over the machinery of the plant, so that now the pathogen's capable of maybe reproducing itself inside of the plant or using the plant to make more of itself for it, which is pretty ingenious but does happen with some pathogens. Or it's just taking over the tissues of the plant, residing inside of it and doing damage at the same time. Then you got to have reproduction. Remember, these are living things in most all cases, these pathogens, and they're looking to reproduce. So you have a reproduction of the pathogen, then the babies, if you will, the offspring, the spores, there's a variety of names, cells, those have to be dispersed out away from that diseased plant to another plant to start this whole process over again. And then you got to think about the fact that that pathogen has to be able to survive between hosts. Some can't. They're obligate. They must have a living host at all times. Others, they can last between growing season. They can, survive in the absence of a host. There are some plant pathogens that can lay in a field, in the soil, in a piece of diseased tissue for 50 years and still be capable of causing disease in a new plant host. Let's talk just a little bit about